Hello guys, how are you? I hope that you are very well today, and this video is for you can see my exposition for Abu Dhabi, and please your comments, please write it on a paper and send me a photo or for email, or I don't know. It's important to review your comments to do a good exposition in this event in Abu Dhabi. And congratulations for the prize on the last Friday. I am proud of you. And okay, we start. Hello, my name is Omar Méndez. I am a student in the La Salle University in Bogota, Colombia, in the Animal Science program. And today I am going to talk about my project, that is a taxometric and nutritionally assessment of 3 New Zealand ryegrass genotypes as a strategy to adapt to climate change in Cundinamarca. And this is a little description of my project. My project was in Colombia, in Cundinamarca. Cundinamarca is the same that Abu Dhabi is a United Arab Emirates, but in Colombia. And Cundinamarca in this moment has affected for the climate change for different reasons. For this, the temperature and precipitation in different places of my country has increased. For this, this project proposed the use of the improved pastures such as a ryegrass, like a strategy to adapt to climate change. For this, the objective of this project is taxometrically and nutritionally evaluate 3 New Zealand ryegrass genotypes as a strategy for climate change in Cundinamarca. And the hypothesis is the Samson ryegrass variety is the best suited to agroclimatic conditions in Cundinamarca. The methodology is very easy. The experimental or the study was in the La Salle University in Bogota, Colombia, in experimental area, where there were 3 treatments, each treatment with the 5 repetitions, and with the 2 types of measures, taxometric measures and nutritionally measures. And the results, ryegrass variety Samson presents a significant difference at the taxometric in the higher height, number of leaf and biomass, and then nutritionally, the variety prospect presents a highest level of crude protein. For this, the conclusion of the project is the Samson variety has reported a higher yields in the 11 taxometric, as well a prospect variety at the nutritionally level with higher potential options. The second part is the introduction. In this part, I review a little more in these items. Okay, my country is Colombia. Colombia is located in the north of South America on the equator line and is a tropical country. In my country there are different animals and a big biodiversity and different life zones. Cundinamarca is a department located in the center of my country, but Cundinamarca and Colombia in this moment had gravely affected for the climate change, But what is the climate change? Climate change is a change in the climate for 2 principal reasons, for natural process and human activities. These maps shows 2 important variables for review the climate change between 2017 until 2018. The first map is a temperature map, and you can see that the temperature has increased in all countries. For example, in the center of my country, where Cundinamarca is, the temperature has increased 1.4 degrees Celsius. And the second map, you can see 2 problems, the north and the south of my country, where the precipitations is decreasing, as opposed to in the center of my country, where the precipitations is increasing. For example, again, in the Andean region, where Cundinamarca is, the precipitations has increased 3.5%. These changes in the climate are results of the greenhouse gases production in general in all Colombia. But this graphic shows 4 different sources that greenhouse effect gases production in the livestock sector. So there are enteric fermentations, manure management, energy consumption, and animal feeding. But please look at the purple part because just this part represents near to 50% of the all gases. Is a result of the enteric fermentations which produces methane gases. For this, the FAO, the Food and Agriculture Organization of the United Nations, proposed 3 ways for reducing or 3 ways to reduce these emissions in Colombia. The first way is productivity improvement that to reduce emissions intensities. The second way is a better livestock integration in a circular bioeconomy. And the third way, where my project takes focus, is carbon capture through improvement pasture management. With this logic in mind, pasture productions review a little more. What is the forage? Forage is a grass or plant that you have for animal feeding. In Cundinamarca, there are 37 million hectares in use for the livestock sector. That of total, there are 22 million hectares just for forest production. That is, in Cundinamarca, there are 464,000 hectares for forest production in specific for milk producers. This makes Cundinamarca one of the biggest milk producers in Colombia, with the 20% of the total nation. In the forage context, Cundinamarca has different types of ryegrass, with the Centrus penicetum and ryegrass or Lolium perenne, that is a scientific name. Ryegrass is a type of grass that can be grown in soils from 2,200 to 3,200 meters above sea level, and also can be grown in soils variability, for example in heavy, fertile, and humid soils. The ryegrass has 2 classifications: chromosomal value and life cycle. In the chromosomal value, there are diploid and tetraploid ryegrass. In the tetraploid ryegrass is a duplication in its chromosomal number for a better agronomic behavior and a better nutritional qualities. But diploids is important for its hardiness. And in the life cycle, there are an annual ryegrass and perennial ryegrass. In the annual ryegrass has a duration of 1 year, and perennial ryegrass has a duration of 5 years or more. And there are another type of ryegrass in these classifications that has a duration of 2 or 3 years. That is intermediate ryegrass. With these classifications, ryegrass has many varieties. But in this project, use Samson Prospect and 150. Why these varieties? Because these varieties, New Zealand varieties, diploid ryegrass, Lolium perenne, and there are another important quality that they're resistant to Roya. Roya is a fungus that can be grown in these plants. For this, the problem question in this project is which of these variety of ryegrass adapts better to the agroclimatic conditions in Cundinamarca. For answer this question, this is the objective. The overall objective is to evaluate the productive potential of the varieties ryegrass, New Zealand, Lolium perenne varieties, Prospect, Samson, and 150 during the establishment and production period, and nutritionality qualities in the agroclimatic conditions in Cundinamarca, Colombia. The first objective specific objective is study the agronomical behavior of the cultivars during the establishment and production period. And the second specific objective is evaluate nutritional potential of the cultivars during each period. For this, the hypothesis of the project is the Samson ribulus variety is the best suited to agroclimatic conditions in Cundinamarca. In my methodology, the methodology has 4 phases: location, sowing and maintenance, data collection on crops, and statistical analysis. The first phase is location. The experimental was in the La Salle University in Bogota, Colombia, in experimental area called a live collection of cool climate forage vegetable species, with an area that 137.5 square meters with a pH that 6.5. The second phase is sowing and maintenance. That this phase there are 2 parts. The first part is a treatments and the second part is distribution. In the first part there are treatments. Each variety was a treatment. For example, the variety prospect was the treatment number 1, and each treatment was marked with a flag. So the treatment number 1 was prospect with a gray flag. The treatment number 2 was 150 with a blue flag, and the treatment number 3 was Samson with a red flag. The second part is a distribution. This area was divided in 2 blocks for a whole, and each block was divided in 8 plots with the infiltration ditch. 4 has 5 repetitions for treatment. The next phase is data collection and crops, and in this part is important to know the periods. The first period is establishment period, and this period has a duration of 63 days, and is that grown the plant until the first cut or the first grazing. And the second period is the production period and has a duration of 28 until 49 days and is between cuts or between grazings. And okay, the measures, there are 2 types of measures. Dasometric and nutritionally measures. In the dasometric, there are biomass, width of leaf, height, number of leaf, and these variables was taken in the file, and the foliar area index was taken with a septometer in the laboratory in the La Salle University. The second type of measures is nutritionally, where there are dry matter percentage, crude protein percentage, acid and neutral fiber detergent percentage, ethereal extract percentage, and ash percentage. This was taken in the animal nutrition in the animal nutrition laboratory in the La Salle University. And the last phase is the statistical analysis. The statistical analysis was random complete blocks analysis of variance to obtain difference between treatments was used the 2K test. And the statistical package used was SAS version 9.2 of 2008.
In the results, again, there are 2 types of results:
dasometric results and nutritional results. In the dasometric results, there are 2 variables that was taken just in the establishment period, and there are another 2 that was taken in the both periods. The first variable is number of leaf, and important before that start in the results, these column graphics are average of total data in this period. And in the second part, in these results, there is a comparison between establishment and production period. But first review these variables that was taken just in the establishment period. Okay, in this graphic there is average. There aren't a significant difference in the number of leaf, but this graphic is a behavior during 4 last weeks in the establishment period in the number of leaf, where there are a significant difference between treatments. the treatment number 3, Samsung, reported the highest number of leaf respect the another treatments. And in the foliar area index, there aren't a significant difference in the average, and this graphic shows the behavior during the establishment period, during all establishment period, and again there aren't a significant difference. Okay. In the height, there are a significant difference just in the establishment period. That have been the treatment number 3 reported a highest height in the establishment period, but in the production period there aren't a significant difference. The last dosimetric measures is width, and there aren't a significant difference in the both periods. The another type of results is nutritional results. And the first variable is dry matter percentage, and the second is ash percentage. But in this average, in these comparisons between periods, there aren't a significant difference. In this poster, you can see the ethereal extract, the fibers, and in these variables, again, there aren't a significant difference in the both cases, establishment and production period. But in the last variable, that is crude protein percentage, and okay, the last variable is crude protein percentage, where there are a significant difference in the establishment period. That the treatment number 1 prospect reported the highest levels of crude protein percentage, next to treatment number 2, Samsung, and the last was treatment number 2, 150. But in the production period there aren't a significant difference. For this, the conclusion of the project is the ryegrass variety Samsung has a better agronomic variables, the first, and the second is nutritionally the ryegrass variety prospect was the best. And it's all. Thank you guys. I wait your comments, your questions, your ATC and no more. I wait your comments and your questions and ATC. Bye.